So Peter Eggenhuizen is a research fellow in the Centre for Inflammatory Diseases at Monash University, Melbourne, Australia, and co-inventor of the immune tolerance platform in the Treg cell therapies laboratory. Using the insight that antigen-specific Tregs could be used to treat autoimmune diseases, Peter developed the sequence of methodologies for receptor discovery and characterization, and engineering Treg cell-based therapies for autoimmune diseases such as lupus, inflammatory bowel disease, IBD, and autoimmune kidney diseases. Peter is passionate about improving treatment outcomes for autoimmune disease patients. So, Peter, thank you very much indeed for joining us today. And the title of your talk is Sm-specific regulatory T cells halt the progression of lupus nephritis. Over to you.
Okay, thank you very much, Jeremy, for the introduction. Systemic lupus erythematosus, or lupus, is an autoimmune disease that affects about 1 in 1,000 of us, but it predominantly affects women of childbearing age. Its pathogenesis is multifactorial and heterogeneous, and it can affect many organs of the body. But it's really the kidney involvement called lupus nephritis that is a leading cause of morbidity and mortality that affects about 50% of lupus patients. As with many autoimmune diseases, lupus also has a dysfunction in the T regulatory cell compartment, or Tregs. And this has been shown by both decreased numbers and defective function. For lupus, the target autoantigens are quite varied, such as double-stranded rDNA, Rho, LAR, histones, U1-RMP, and the Smith protein. And it'll be the Smith autoantigen that I focus on in this discussion. So the Smith proteins are a group of RNA binding proteins that are expressed in all cells. And the Smith protein is the autoantigen that's most specific to lupus, with the other autoantigens also being found in other autoimmune diseases. So why is the Smith autoantigen important? Well, there's been evidence to show using animal models that in both rabbits and HLA transgenic mice that were immunized with the Smith peptides, that they developed lupus-like nephritis disease. As well, when looking in lupus patients, they were able to identify clonally expanded Sm-specific T cells. And also having autoreactivity to the Smith antigen is associated with both early and poor prognosis in lupus nephritis. And also knowing from our previously published work that it's specifically the autoantigen-specific Tregs that are lacking in autoimmune disease patients, that we aimed to develop a Smith-specific T cell receptor, or TCR Treg, with the aim of restoring immune homeostasis and halting lupus nephritis disease progression. So to achieve this, we set out on a platform of development. The first starts with identifying the immunogenic Smith epitopes specific for HLA-DR15, which is the haplotype that's most associated with lupus nephritis. We then move on to identify T cell receptors that are specific for that epitope, characterize them, and choose a lead TCR sequence that we then validate before moving on to GMP cell manufacture and a clinical trial to test safety and efficacy. So the first step in the journey is to identify the immunogenic Smith epitope. We first started out by using ProImmune's Reveal MHC peptide binding assay that used HLA-DR15 as the substrate, and then interrogated it with overlapping 15mers of different Smith proteins. And as you can see in the SMB/B prime protein, there's a hotspot region of binding to HLA-DR15 around epitope, around amino acid position 20 that had a both highly stable and long binding, as opposed to the rest of the epitopes of the Smith proteins that were not as strong. And as you can see down the bottom, there's just a summary of the best binding epitopes of the Smith protein for HLA-DR15. So using this information, we did an in vitro proliferation assay where we took the top 3 candidates from the proimmune reveal assay and looked at the cell trace violet dye dilution to indicate T cell, CD4 positive T cell proliferation. And it was the SMB/B prime 58 to 72 peptide that induced the highest amount of T cell proliferation. And we used this peptide moving forward, and I'll just call it the Smith epitope from now on. In our lab, we also identified, solved the crystal structure for the Smith epitope bound to HLA-DR15. And using this information, we then single-cell sequenced the proliferated CD4 positive T cells in the presence of the peptide, Smith peptide, and identified T cell receptors specific for the Smith peptide. We found that there was a highly proliferated T cell receptor clonotype called TCR1, followed by other also highly proliferated T cell receptor clonotypes. And it's TCRs 1, 2, and 3 that we then made lentiviral vectors out of and transduced first T cell line called Jurkat cells. So using the TCR transduced Jurkat cells, we were able to do a TCR binding affinity assay using limiting dilution of antigen-specific multimer, which derived a apparent affinity of the TCR for its ligand. And we found that it was TCR1 that had the highest apparent affinity, followed by TCRs 2 and 3. Further using these TCR Jurkat cells, looking at the immune synapse by imaging flow cytometry, we were able to see that specifically at the immune synapse, which is the point at which the peptide MHC binds to TCR, in the presence of the Smith peptide, we were able to see an increase in T cell activation by activation markers CD3 and filamentous actin. And this was seen across both TCRs 1, 2, and 3. So to choose our lead TCR moving forward, we selected TCR1 based on highest apparent affinity and highest clonotypic abundance. We then used this lead TCR to transduce primary human Tregs. And by flow cytometry, we were able to see about 20% of the Tregs successfully transduced with the TCR. And there was of the Tregs that were transduced, about 90% of them co-expressed GFP and TCR. These TCR Tregs, when transduced and expanded 10 days in vitro, showed a stable Treg phenotype of 98% CD25 high, CD127 low negative, and also high expression of FOXP3 and Helios. Further looking at the stability of these Tregs once transduced and expanded longer term in culture, we were able to show that at the FOXP3 TDSR region, the methylation status there, what at 3 weeks transduced, was similar to freshly isolated day 0 untransduced Tregs, suggesting that the phenotype of the Tregs is relatively stable over time in culture. Also looking at pro-inflammatory cytokine secretion of the day 10 expanded and then PMA inomycin stimulated cells, the SMTCR Tregs produced a low level of both IL-17A and interferon gamma. Further characterizing the functionality of the SMTCR Tregs, We performed activation assays using T cell activation marker CD69 and the Treg specific activation marker GARP. And we saw a dose-dependent activation response when the Tregs were transduced with the SM-TCR, but not in the mock TCR Tregs. We also did a suppression assay to look at the suppressive capacity of the Tregs over the T conventional cells in the well. And when the SM-TCR Tregs are present, there was a superior suppression of the T conventional cells compared to mock Tregs. So overall, we're on our pathway to creating a cell-based therapy that could be used to treat lupus nephritis patients. We're up to stage 4. We're still working on the validation of the lead TCR in both in vitro and in vivo work. And then leading on from that would be GMP cell manufacture and hopefully in the not too distant future, a clinical trial. So thank you to very much to everyone who's been involved in this project over the years. And if you're interested to read more, it's also just published in Nature Communications. And thank you for listening.
Right. Thank you very much indeed, Peter. That's really fascinating. I have maybe a question just to kick things off with. So obviously you were focusing on that one specific, I think 58 to 72 epitope predominantly, based on the T cell proliferation data, which obviously makes lots of sense. Is there any particular value in looking at TCR specificities from some of the other epitopes, or were you really just trying to kind of demonstrate proof of principle with the sort of the dominant T cell response that you were seeing? I'm just wondering whether you did any additional studies with some of those other epitopes that you'd identified in the reveal assay and the subsequent T cell proliferation assay as well.
Yeah, we really focused on just the one, but we do have the data, and we're looking into it for the other ones that could be used as a co-therapy potentially, or yeah, we do have them, and it is possible to also
Mhm.
include them.
So I'm just wondering, do you think that as a principle, it would actually be of benefit to have that variety of TCR specificities rather than just having it monospecific?
Yeah, I guess it's really hard to know without doing a clinical trial how it would work in patients. But definitely it could be possible to do a multi-approach. It just would involve yeah, finding a good TCR again against the other epitopes and characterizing that.
Yeah, great. No, thank you. Questions, anybody else? It's not just me, I hope. So feel free to put your hand up there. If you're not sure where to go, you can go to the chat function at the bottom of the screen and you can type in a question. And then there's also an opportunity under reactions. There's a reactions button. You can click on that and then you can just raise your hand and then we can see who has any questions.
I just had a quick question. Um, just in terms of applicability for other autoimmune, it seems like this would be pretty easy to kind of plug and play this approach into other disease indications. Are there other kind of low-hanging fruits that you might be going after next?
Yeah, there's a few other autoimmune diseases we have in that pipeline that I showed. Um, so yeah, obvious ones are type 1 diabetes. Goodpasture's disease is something that our lab does commonly. And also I mentioned inflammatory bowel disease, rheumatoid arthritis, Sjogren's syndrome. Yeah, it's like you mentioned, it's endless, and it's just a matter of yeah, thinking about the low-hanging fruit or the unmet need for these patients, which ones we choose.
Thank you.
So another question, Peter, I have is just in relation to the HLA restriction, because obviously, again, the focus was on DR15 due to the association. Are
Yes.
there any other associations in lupus, or is it really very strongly just DR15?
Yeah, it's predominantly DR15 and DR3.
Hmm.
So I only showed the data for DR15, but we're also looking into DR3 in the same way. And that has a quite a high coverage of lupus nephritis patients. We're particularly focused on those patients because those are the ones with the most severe disease.
Great, thank you. And then just in regards to the clinical approaching, so the aim is to I think I missed it, so apologies for that, but you're saying that you're just doing autologous T cell transduction with lentivirus
Yeah,
transduction. Okay, yeah, that's the
yeah.
plan. Cool. And what stage have you got to in that regard?
We haven't started the clinical trial yet. We're about 2 years away. We just need to finalize our preclinical data set,
Mhm.
and then, yeah, move into
Yeah.
clinical trial.
Well, good luck. Look forward to hearing the next chapter. We really appreciate it's nice to have a story from the start of the discovery point to where you've got to now. So thank you very much for particularly for staying up this late in the day where you're on the other side of the world. So we really appreciate it. Thank you very much, Peter. Are there any other questions from anybody? I can't see any hands up. So we'll probably move on to our next speaker. So yeah, thanks again.